Welcome, everyone, to the final webinar of this series, the 2024 AGM Preclinical Translational Science webinar series. We're super excited today to have Dr. Bryan Roth with us. Dr. Roth is a professor of pharmacology at UNC School of Medicine. He has published more than 500 papers and given more than 300 invited talks. He is an elected member of the National Academy of Medicine and the American Academy of Arts and Sciences. His work focuses on chemical and synthetic biology. And today he's going to talk to us about drug discovery meets AI. We're super excited to have him. I'm sure you'll have plenty of questions. So if you have any questions, please go ahead and record your questions in the Q&A box. And we will get to those questions at the end of the talk. Bryan, on behalf of my colleagues here at NCAT, everyone on attendance today, we're super excited to have you. The floor is yours.
Thank you. Can you see the screen?
Yes.
Okay, great. Excuse me. So thank you very much for this invitation to speak here. What I'd like to do is basically tell you a couple of stories here. First, a conventional drug discovery story, and then how we're trying to accelerate drug discovery for serious neuropsychiatric diseases by using advanced computational technologies. These are my disclosures. I'm going to talk a lot about this compound BMB 101, which is now in phase 2 clinical trials. Lots of our technology has been licensed by biotech, and all the work is supported by the NIH or DARPA. What I'm going to do today is talk about serotonin and serotonin receptors as targets for drug discovery. And of course, serotonin, as you can see here from this tattoo, not my tattoo, something I found on the web, is an indole amine or a biogenic amine neurotransmitter. And the cell bodies in the brain for serotonin are found here in the raphe nucleus, and they project widely throughout the brain. And serotonin has been implicated in virtually all behaviors and functions of the brain that are listed on the left, as well as many effects outside the brain, including regulation of multiple organ systems. And there are a number of medications that you can see there that interact with serotonin receptors to mediate their actions. Now, to mediate the effects of serotonin, a family of 7 receptors has evolved, denoted 1 through 7, as you can see here. And today we're going to focus on the 5-HT2 family of receptors. So there are 3 members of this family. They're all G protein coupled receptors. We'll talk a little bit about what those are here shortly. But these include the 5-HT2A, the 5-HT2B, and the 5-HT2C. And the 5-HT2A receptor is primarily responsible for the action of drugs that are psychedelics, which activate the receptor, and many atypical antipsychotic drugs, which are antagonists at the 5-HT2A receptor. So you have this interesting situation where drugs that activate the receptor cause hallucinations, and drugs that block the receptor block hallucinations in humans. The 5-HT2B receptor has gained most attention because drugs that activate this receptor cause valvular heart disease in humans. So it's a key off-target to avoid. And the 5-HT2C receptor is essential for the action of certain anorectic agents, as well as new drugs which are being advanced, which have the potential to be really transformative in terms of treating seizure disorders. The 5-HT2 family signaling pathway is relatively complex. As I mentioned, these are G protein coupled receptors, 7 transmembrane domains. Following agonist binding, there is an exchange of GDP here for GTP in the heterotrimeric G protein. And this then causes the dissociation of the heterotrimer, and both the alpha and the beta gamma subunits then activate various downstream effectors. In this case, these are all coupled to phospholipase C beta, which ultimately releases inositol trisphosphate to release intracellular calcium, and diacylglycerol, which activates protein kinase C. So this is sort of canonical GPCR signaling. This is the pathway that these receptors canonically use. Following this, there is receptor phosphorylation by a number of different kinases, including G protein receptor kinases. This potentiates the binding of arrestins, which have two functions, or many functions. They block the ability of the receptor to interact with G proteins, hence they arrest signaling. And they also serve as scaffolds for various downstream effector systems. And it's thought that signaling through arrestins can be different in both space and time and texture from signaling through G proteins. And my lab has spent many decades studying the intricacies of serotonin receptor signaling. And this is a review you can read about it. So some years ago, now about 20 years ago, we were interested in developing drugs that are selective for various serotonin receptor subtypes. And we, for reasons that I won't go into now, we executed a high throughput screen of, let me just close something here. A high throughput screen of 5-HT2A and 5-HT2B and 5-HT2C receptors using basically a flipper in the 384-well format. And we identified this interesting drug, tranylcypromine, which appeared to be a selective 5-HT2C agonist. That structure is shown there. Now, tranylcypromine was interesting to me. So one of the hats that I wear is I'm a psychiatrist, and for many years I treated individuals with severe psychiatric illnesses. And it was known to me and others that tranylcypromine is a monoamine oxidase inhibitor, but unlike other monoamine oxidase inhibitors, which are commonly used, it causes weight loss in humans. All others cause weight gain. And we figured this might be because of off-target effects as an agonist at the 5-HT2C receptor. So we decided to use this compound going forward for a medicinal chemistry campaign. And one of the first derivatives that was created, and this was a longstanding collaboration with the Alan Kozakowski lab, which was then at the University of Illinois, Chicago. This compound C37 arose. You can see it differs from the parent compound by 2 methyl groups. And in vivo assays for potential antidepressant drug-like activity, you can see it has sort of modest activity here compared to the positive control sertraline. This is a typical tail suspension test. These were done by Bill Wetzel's lab at Duke. Even though it had rather modest activity, we're encouraged that it had basically any activity at all, and then extensive focused medicinal chemistry was done by the Alan Kozakowski's lab, and John McCorvey from my lab led the pharmacologic characterization. And literally hundreds and hundreds of analogs were made and synthesized and characterized in vitro and in vivo assays, ultimately leading to this compound here, which disclosed not only antidepressant activity, but potential antipsychotic activity in various mouse models. And the interesting this compound ultimately was through the support of the NIH, actually, IND enabling studies were done, and this compound and relatives ultimately were licensed to Bright Minds Biosciences in 2021. It was advanced to phase 1 clinical trials for Dravet syndrome as an anti-seizure agent in August 31, 2022, successfully passed those trials. And phase 2 trials for a number of serious and intractable seizure conditions have now been announced by Bright Minds Biosciences in 2024. So you have this, and the thing that was remarkable about these compounds, and this was basically the way they were designed, is because they're agonists at the 5-HT2C receptor, the concern, particularly in treating seizure disorders and other chronic conditions, is that agonists tend to lose their potency over time. This is called desensitization. And the main pathway responsible for desensitization is this arrestin pathway. And so what we did is we were able to identify actually compounds that are super agonists. This is one that's just a full agonist, but we actually have compounds that are super agonists for mobilizing intracellular calcium and activating GQ pathways, and are weak and in some cases inactive at the arrestin pathways. So these are G protein biased, and they show greatly attenuated desensitization. And this will be a distinct advantage for these compounds going forward in clinical trials. And you can see that this compound has a very high bias factor there for those that are interested in that. And the lessons that we learned briefly over 20 years was that there was this evolving utility of the lead compounds. Initially, antidepressant compounds, then antipsychotics, and finally anti-seizures. And this was identified through actually a zebrafish screen that was done by European colleagues. And all the therapeutic indications are on target. They're due to the 5-HT2C receptor. And the key thing here is that it has minimal activity at the key anti-targets, which are the 5-HT2A. Of course, you want to avoid agonism of that because such drugs are psychedelic, and the 5-HT2B receptor because such drugs cause valvular heart disease. And so this was 20 years of focused pharmacology and medicinal chemistry, and successful licensing was the ultimate key to clinical development. But I just like to point out that everything up to licensing was actually supported by the NIH. So it was a nice win for public drug discovery. This was, this took a really long time, and at the time it was unknown how selectivity and bias signaling were attained. And the question we wanted to address next is, will structures be helpful? And the idea is that if we actually had structures of these receptors, maybe this would help us for structure-guided drug discovery. And before I get into this, I want to discuss our efforts in creating safe and effective drugs that interact with the 5-HT2A receptor that are not psychedelics. And the reason for this is that psychedelics, of course, are proving effective in treating many mental health and substance use disorders. And classic psychedelics are all agonists at the 5-HT2A receptor. These include drugs like LSD. Psilocybin is actually a prodrug of this drug, psilocin. Mescaline from peyote cactus, and DMT. And as I mentioned, there now are a large number of phase 2 clinical trials showing that either a single or multiple doses of psilocybin, for instance, appear to be remarkably effective for treatment-resistant depression. And they have basically raised the possibility that psychedelics might prove to be transformative treatments for a number of neuropsychiatric disorders. Of course, the problem with rolling such compounds out to the general population is they're psychedelic. And this can be a serious problem for many individuals. The other problem with these compounds is they have the parent compounds extremely complex pharmacology. So this is a little diagram we put together. This is a phylogram of the G protein coupled receptors in the human genome. And what I've indicated here in the red dots here are all the receptors that LSD activates. And that's from this paper here. And despite its agonist activity at probably a couple of dozen different receptors, it's pretty clear that the psychedelic and potentially therapeutic actions are mediated only by a single receptor, which is the 5-HT2A receptor. And we wondered if this complex pharmacology was shared with other psychedelics. And in a large series of studies, which we're currently preparing for publication, we screened all, not all, but most of the known and validated psychedelic drugs that have been used in humans, along with many non-psychedelic chemical analogs, and screened them using the resources of the NIMH Psychoactive Drug Screening Program, which I run. And you can see that from the heat map, all of these drugs have a very complex polypharmacology. And the challenge here is that there's only a single receptor here, the 5-HT2A receptor, that we believe is responsible for both the beneficial and the deleterious actions of psychedelic drugs. So the question is, how can we make drugs that selectively activate that receptor and don't cause a psychedelic experience? We want them to only have a therapeutic effect. Now, the evidence for 5-HT2A receptors as targets of psychedelics come from studies that have been done over the past 40 years. Initially, studies in Richard Glennon's lab showed that the psychedelic actions in mice correlate with the serotonin receptor affinity. Studies in knockout mice show that such mice do not respond to psychedelics. This is our particular paper here in Nature. But the best data here were studies from Franz Vollenweider's lab that have been done decades ago, showing that in humans, the relatively selective 5-HT2A antagonist ketanserin essentially blocks all of the psychedelic effects of psilocybin, LSD, and recently reported mescaline. So the actions are mediated through the 5-HT2A receptor. Now, the consequences of this pharmacology is one of the key concerns that we've had is that psilocin and LSD are very potent 5-HT2B serotonin receptor agonists. And of course, drugs that activate these receptors cause valvular heart disease in humans, something that was discovered by my lab in the early 2000s. And many drugs with structural and pharmacological similarity to LSD also cause valvular heart disease. For instance, chronic use of ecstasy, or MDMA, has been associated with valvular heart disease. And it is unknown which of the many additional receptors targeted by LSD, psilocybin, and so on may be associated with untoward medical consequences or therapeutic actions. Now, our current understanding of psychedelic drug action is that, as I mentioned, it's mediated through the 5-HT2A receptors. If we focus on psilocybin, the active ingredient of psychedelic mushrooms, psilocybin itself is a prodrug. The psilocybin itself is inactive. It's dephosphorylated to psilocin, which then interacts with 5-HT2A receptors, which are found on the apical dendrites of layer 5 cortical pyramidal neurons, something my lab discovered many years ago. This leads to a very complex signaling cascade, which we continue to study, ultimately leading to increased excitability of the neurons and increase plasticity. And the very first demonstration that psychedelic drugs cause increased plasticity was by Jones et al. This was a collaboration that my lab was involved with the Peter Penzis group in 2009. And it's thought that that these plasticity-inducing actions of psychedelic drugs are ultimately involved in their therapeutic actions. Now, my lab has focused many years on on understanding how it is that drugs like LSD, psilocin, and others directly interact with the 5-HT2A receptor. Again, with the hope that once we understand their structures, we can use this information for structure-guided drug discovery. This is just a little animation I'm going to show you, which is based on many papers we've published and was prepared by Gabrielle Ashelman of Ribosome Studio. And so this is LSD. You can imagine somebody has just ingested LSD. It's floating through their brain. And in the background here in white, you can see the 5-HT2A serotonin receptor. And I always like to point out the large extracellular amino terminus of the 5-HT2A receptor. Still don't know what the action of that is. Now, LSD itself interacts in a complex way with the receptor, ultimately stabilizing the active state of the receptor, hence the light coming from the receptor. Of course, there's no light in the brain. And then this leads ultimately to the dissociation of the heterotrimer. You can see the alpha subunit here in purple and beta gamma going off into the distance. And here, these flashing lights here denote intracellular calcium. So we think the final signaling is actually mediated by changing the electrical properties and the plasticity properties through this canonical G protein pathway. And so the question is, can we use these structures directly for drug discovery? And this is where we get into the main message that I'm going to present today, where we use computational approaches, not really generative AI, but you could call them AI-esque approaches. And the key feature of the approach that we use, and this has been used in collaboration with the Irwin and Choiquette labs at UCSF now for almost 2 decades, is we first, initially we would create a model of a receptor. These days we just solve the structure of the receptor. And then the structures are used directly for drug discovery. And we use computational screening, primarily docking, which I'll explain in just a minute. But the key advantage of this technique compared to physical screening, which I know NCATS is sort of specialized in, is we and others have found the hit rate for physical screening to be at best 1 in 1,000. And actually, if you have a hit rate greater than 1 in 1,000, you probably have problems with your assay. Whereas the hit rate for virtual screening is, in our hands, between 5 and 40%. So this is 50 to 4,000-fold better. So this is really accelerates drug discovery. And of course, by virtual screening, we can screen billions or trillions of compounds, more than could ever be screened by physical approaches. And the approach that's taken, and this was pioneered by the Irwin and Choiquette labs at UCSF, is to take libraries of available molecules, which are enumerated in silico. And these libraries now are in the billions of compounds. And then these are docked at the molecular target in multiple conformations. A key feature of their approach is that each is docked in at least in up to 1 million different orientations and conformations. They're scored for fit, and then the highest scoring molecules are then synthesized. Of course, these molecules do not exist in the physical universe. They're only molecules that, in theory, could exist. So they're synthesized and ultimately tested. Occasionally I'm asked about the docking scoring functions. This is, again, been pioneered by the Choiquette and Irwin lab. And I urge you to read their papers to find out about more about this. But this was sort of the very first demonstration of how successful this could be. In 2016, we were engaged in solving structures of GPCRs. This was a high-resolution structure of a particular dopamine receptor we solved, the D4 receptor. with the atypical antipsychotic drug nimonipride. This was a very high resolution structure, 1.9 angstroms, allowed us to visualize not only sodium, but also all the water molecules that were interacting with the receptor. And we found actually that nimonipride was stabilized by an interaction with a water molecule in the extracellular loop. In examining the structure here, let me just go back here just a second here. There we go. In examining the structure, so once we had obtained the structure of the D4 receptor, we and others had obtained simultaneously structures of all the other dopamine receptors. And what we noticed about the D4 receptor was that there was this pocket here, which was not seen in the D2 receptor, which we had and actually published a year later. And we wondered if we could use this, this new pocket for a focused docking effort to identify compounds that are selective for the D4 receptor. So here's the pocket. You can see this extended binding site here. And so what Anat Levitt did was to perform virtual screening with several million compounds. High-ranking compounds were tested. This was the initial docking hit. They were then optimized by scaffold hopping and what's called analog by catalog. And in each round of optimization, compounds were tested, and further scaffold optimization was performed. And ultimately, we obtained this compound here, 924, which turned out to be a selective D4 antagonist. And that was published in Science some years ago. Now, we later, through the resources of the Druggable Genome Initiative, were able to increase our ability to do docking. This is one of the successes here. We performed the first ultra-large scale docking campaign against a G protein coupled receptor. This was published in collaboration with the Choiquette lab in 2019. And just out of the docking screen directly, without any further optimization, we were able to obtain a picomole potency, highly selective, G protein biased D4 agonist. So this tells you the power of this particular approach. Now, as we were developing our computational and physical screening pipeline for G protein coupled receptors, other computational technologies arose, including AlphaFold, and it was wonderful to see AlphaFold, the AlphaFold group, win the Nobel Prize this year. So I guess I don't have to explain to anyone what AlphaFold is. Interestingly enough, for all of you who are not computational biologists, when I would present this work before they won the Nobel Prize, most people had not heard of AlphaFold, and I guess everybody has now. But at any rate, AlphaFold is a quasi-artificial intelligence system which provides fairly accurate protein structure prediction. And they've been able to now basically any amino acid sequence now a structure can be predicted from. And we and others have wondered if AlphaFold could provide a template for drug discovery. As I mentioned, in the past, we used primarily structures or models that we had created for docking campaigns. But the question is, could we use these AlphaFold models directly without going to all the trouble of solving structures? And this was a project which was begun some years ago by J.K. Liu and Nick Kopalka in my lab, J.K. from Bryan Choiquette's lab. And what was done was we solved a new structure of the 5-HT2A receptor bound to the non-psychedelic compound lissuride, because our goal was to make compounds that are not psychedelic. And this was a structure that we did not deposit in the public domain so that it could not be used for training of AlphaFold. And then we took the AlphaFold prediction for the 5-HT2A receptor and compared them. And you can see that The overall three-dimensional organization of the receptor and the model are quite similar, but there were key amino acids here that are in the binding pocket, which are in orientations which would not allow conventional 5-HT2A agonists to bind to them. And so the model appeared to be wrong. And so we were skeptical, actually, as to whether the model would provide a reliable template for identifying compounds. Now, in the past, a number of papers have appeared with basically this same conclusion, that AlphaFold gives a pretty good estimation of the overall orientation. But if you look at sort of key residues and active sites, they're frequently in the wrong orientation. And AlphaFold is just not able to retrospectively identify known compounds that interact with the binding sites. The question we had, though, was perhaps AlphaFold is generating alternative conformations which we ordinarily don't see. And if we screened the AlphaFold models prospectively, they might be useful for drug discovery purposes. And so this was the experiment that we set up. We by meaning JK, performed a docking screen of 1.6 billion molecules against both the cryo-EM structure and the AlphaFold2 structure. And from this, around 200 compounds from each bin were synthesized and tested by Nick in my lab. And this is a waterfall plot here for both campaigns, and you can see that compounds that arose from the structure or from the AlphaFold2 prediction had basically the same hit rate, about a 25% hit rate. And the number of high quality hits here, what we are ultimately looking for are G protein biased agonists, was relatively similar for both campaigns, which was I would say really surprising to us. And one of the big surprises was that there was absolutely no overlap in the chemotype identified from both docking campaigns. So what we found is we have this very interesting situation where the experimental structure and the AlphaFold model appear to work more or less equivalently in ultra-large scale virtual docking campaigns, but that they identify distinct universes of small molecules. So one of the advantages of this then is it basically doubles the chemical space that you have to choose from in ultimately deciding what compounds you might want to move forward for optimization and ultimately clinical trials. The other thing that was most surprising was the best compounds that we found were actually discovered by AlphaFold. So these are three exemplar chemotypes. You can see all three have distinct chemotypes. And in all three cases, we had drugs that are selective for the 5-HT2A receptor with minimal activity at the 2B and the 2C receptor. And all three are GQ biased compounds. And all three are worthy, actually, of advancing for further clinical development. The big question we had, though, is why is it that this worked at all? And I'm going to show you why we think here, what we think might be going on. So this is if I just go back one slide here, let me go here. So this is the best compound here, 7757, which you can see is essentially absolutely selective for the 5-HT2A receptor, has a modest degree of GQ bias. And we are wondering how it is that AlphaFold identified this compound. And so this is the docked compound to the AlphaFold model. You can see we're going to have a close-up of the binding pocket. I want you to focus here on this tryptophan 151. It turns out this is key. So this is the model of the compound. We then, by we, I mean Ryan Gumper and Uliana Panova and others from Yorgos Skiniotis' lab, we were able to obtain high-resolution structures of this compound with the 5-HT2A receptor, and that's shown here. You can see the structure of the compound there. And again, note that tryptophan here. So that's the structure, actually. And here is what I'm going to show you now is the overlay. So you can see the AlphaFold prediction versus the structure actually were pretty close. Just a difference here in the orientation of the tetrahydropyridine ring. And we think this is due to the differential orientation of this key tryptophan, which we found actually is quite mobile, depending on which particular structure we look at. So in more detail, here's that tryptophan. Here's the AlphaFold model. You can see it's bending over. And here it's sort of lifted up a bit. And that provides just a little bit of wiggle room here for that tetrahydropyridine ring to move up there. And so we think that's the key driver of the interaction. And we think that one of the things that AlphaFold is doing is sort of discovering these intermediate conformations that can be used directly for drug discovery. Okay. Now the question is, so that's a really nice demonstration of how AlphaFold can be successful. I'll just, in all honesty, it's only successful for about 50% of the targets that we work on. Really for understudied G protein coupled receptors for which there's not a lot of structural information, we've not found it to be very effective at all. The other question, though, is can other computational tools accelerate this structure-guided drug discovery? And I'm going to end with a short story done in collaboration with the Drawer Lab at Stanford University. And this was carried out primarily, all the computational work was carried out primarily by Carl from Ron Doerr's lab. And as I mentioned, selectivity, of course, is a long-standing challenge in drug discovery. And as has been mentioned numerous times, selectivity drives desired effects, and non-selectivity typically drives deleterious effects. And for psychedelic compounds, of course, all known psychedelics are very potent 5-HT2A agonists. We think these are responsible primarily for their benefits for neuropsychiatric conditions. But they also activate the 5-HT2B receptor. Every single one of these does. And this is going to be problematic going forward, particularly in cases in which these drugs may be used chronically. And of course, this is something my lab has had interest in, for many, many decades. And the problem that we ran into, so over the years, we've solved a number of structures of the 5-HT2A, the 5-HT2B, and the 5-HT2C receptor. And what we found is basically the binding pockets are essentially identical. So this is an overlay of the 5-HT2A and 5-HT2B binding pockets. You can see the key residues are essentially identical. And this creates major difficulties for structure-guided drug discovery because you don't have residues that you might want to target for selectivity. So this was the approach that they took. This was, I'd say, really creative. So what Carl did were long-term molecular dynamics simulations of initially LSD with the 5-HT2A and the 5-HT2B receptor. And what he found, if you look at it here, was that LSD in simulations transiently adopts this conformation here where a portion of it is pointing to this little cavity between these two transmembrane domains. And you can see that there. Whereas for the 5-HT2B receptor, it was basically locked in this conformation. It didn't move at all. And so this is, if I go back here, this is what could be called a cryptic pocket. So this is a pocket which appears only transiently and is only visualized by molecular dynamics simulations. And so it was then a fairly simple matter to identify the residue here on our exemplar compounds and basically just to insert bulk. So basically methyl and ethyl groups were inserted here. The idea was that they would engage this cryptic pocket. This would repel their ability to interact with the 5-HT2B receptor, and magically we would obtain selectivity. And in fact, it worked extremely well. So these are three parent compounds of different structures. I apologize for not showing the structures here. The paper is currently under review, and we're waiting for it to be accepted before we reveal the structures. But in all cases, we were able to maintain 5-HT2A receptor activity, particularly agonism, and completely abolished the agonist activity at the 5-HT2B receptor. And this, again, is with three distinct chemotypes of three distinct chemical series. So this really works. The question was, are these designed antagonists useful for anything? We had designed them to be partial agonists with G protein bias. And so our prediction from many prior studies was that these compounds would not have psychedelic-like activity, at least in mice, but potentially could have therapeutic utility as antidepressants. And to test this, we collaborated with the Bill Wetzel lab. These studies were done by Ramona Rodriguez and her tireless crew of undergrads and technicians, using a genetic model of depression and antidepressant drug action that was developed by Bill's lab. And basically, wild-type mice here you can see have a high score in mobility. This is a tail suspension test, and by contrast, the mutant mice show greatly reduced mobility. So this would be quote unquote a depressive-like phenotype, but it's not really depression, but serves as a nice marker of antidepressant drug action. And so you can see that the sort of conventional antidepressant fluoxetine, which is known as Prozac, has a pronounced antidepressant effect in these mice, as does psilocin. And interestingly, all our three compounds also have the desired beneficial antidepressant drug-like effect in vivo. And this was acute studies. Now, one of the amazing things about our compounds is they not only have this acute action, they also have a long-lasting action. So after a single dose of the drug, the mice were then tested 14 days later. And you can see that the antidepressant effect of a single dose of Prozac and a single dose of psilocin have basically been attenuated. But our compounds still have this robust antidepressant effect. So these designed agonists induce long-lasting antidepressant-like effects in vivo. So the take-home message here is that ultra-large-scale docking and molecular dynamics, which are the computational approaches that we use, are useful platforms for the discovery of novel chemical matter for molecular targets. We published many, many papers on this in collaboration with computational chemists. And the other take-home message is we're currently there is I would say a revolution in structural biology because of cryogenic electron microscopy. And this affords the opportunity to obtain structures for truly structure-guided drug discovery. And of course, my lab has solved many of these structures that we're using as templates for neuropsychiatric drug discovery. And finally, this approach has led to new medications, both psychedelic and non-psychedelic, targeting this 5-HT2 family of receptor. Currently, our 5-HT2C candidate compounds are in phase 2 clinical trials for a number of seizure conditions by Bright Minds Biosciences. And we're currently identifying the clinical candidates for the 5-HT2A receptor to initiate formal IND enabling studies and to get these compounds in humans. And just a personal note. So the work on the 5-HT2C receptor, as I mentioned, took about 20 years, whereas this work on the 5-HT2A receptor that I summarized here is basically 4 years of work. So it's considerably faster. And this is the team. A number of talented postdocs in my lab have contributed to this. I want to highlight Manesh, who's done just a phenomenal amount of work on the structure and the pharmacology of the hallucinome, and he'll soon be looking for a faculty position of his own. All the other postdocs basically have gone on to lives of their own. I want to give a shout out to Ramona and Bill Wetzel at Duke. John Jin's lab out at Mount Sinai does all the focused medicinal chemistry here that was involved in the computational studies. Brian Choquette's lab leads the docking. Carl did the molecular dynamics simulations. Yorgo was at Stanford, now at St. Jude, has been extremely generous and kind, sharing cryo-EM capability and sort of teaching us how to do cryo-EM. And I really want to thank him and Jimena and Juliana for helping us. And of course, the Alan Kozakowski lab created all of the BMB compounds. And all the work is supported by the NIH and by DARPA. I want to give a shout out to the NIMH Psychoactive Drug Screening Program. Has been absolutely key for everything that we do related to drug discovery. And just to let you all know that this is a free service that my lab runs for the scientific community. Anyone can, any academic or someone who works for a nonprofit can send us compounds, and we're happy to do the off-target profiling for you. This is not only free, it's confidential, and we claim no IP, so it's IP neutral. And I also want to give a shout out to the Illuminating Druggable Genome Initiative. This was quite helpful, NIDA, NIMH, and of course, DARPA. And I think that gives me like 10 minutes to answer questions, so I'm happy to do that at this time.
Thank you so much, Bryan, for a wonderful presentation. There are plenty of questions, so I'll moderate it to help you here. So I'll start with
questions. Yes.
Great. Let's start.
That's good. That's a good thing. Okay.
Yep. All right. So I'll start with this one because it has people liked it the most. So how do you verify receptor occupancy in vivo in the CNS for your projects? And what is your strategy to optimize CNS PK?
So it turns out our compounds have marvelous PK properties out of the box. And that's, if you look at the supplementary data in the papers we've published, it shows that. So our typical compounds have a brain to plasma distribution of greater than 2 to 1. So they have a greater brain penetration than they do plasma presentation. And they're all really small, they're because they arise from docking campaigns, they're all pretty small molecules. In many cases, for a typical medicinal chemist, they would be considered fragments. And because of this, they have really excellent drug-like properties. So we actually have not had to have any, cross my fingers, knock on wood. We've not had any problems with CNS penetration or really metabolic problems either. We have lots of computational screens for those. Ideally, so for the 5-HT2A project, there are a number of PET ligands that one can use to demonstrate or measure receptor occupancy. We don't have those for the 5-HT2C project, but we're confident based on the PK properties that we have 100% occupancy.
Oh, that's good to hear.
Yeah.
Thank you so much, Bryan. All right.
It's just luck, I would say. We haven't. That's something. That's a a bridge we have not had to cross with our compounds.
Awesome. Okay. I'll take this question's next question here. You mentioned screening 1.2 billion compounds to, in the end, arrive at a small handful of active chemistries associated with a single target.
Right.
Future development of these will certainly reduce this to maybe one or two that may go to the clinic. There are thousands of disease targets out there. Considering the 1.2 billion to one campaign, maybe for each target, do we really have enough chemistries to peruse, even in silico, to find all of the active regions of chemical space effective against all of the disease targets we are going after? It's a huge question.
No. That's a great question. Who asked that? That's a wonderful question.
The name is not very clear, so
okay.
yeah, sorry.
Chemical space is essentially infinite. So I left out a slide, but the number of drug-like molecules that could exist theoretically is 10 to the 63rd, which is more than the number of molecules that are known in the known universe. Chemical space is infinite. The advantage of the approaches that we take, which use physics-based scoring functions, is that they don't, unlike conventional sort of AI programs, they don't rely on prior information for predicting structures that bind to the target. They use, each compound is analyzed de novo. So it's this is an approach which basically doesn't suffer from the typical problem of AI or in silico or machine learning based approaches, in that the training set is more or less irrelevant. So the problem with typical machine learning and AI based approaches is it's all dependent on the training set. And of course, in GPCR structural biology, our training sets are quite asymmetric. So we've solved many structures of GPCRs, at least for half of them, we don't have any structures at all, for instance, and have no chemical matter. And so if you use those for training sets, it's basically an asymmetric training set. The advantage of the physics-based approach is that it's training set agnostic. So in theory, it could be used for any molecule that you could enumerate in the physical universe.
I see. Thank you, Bryan. There's two follow-up questions to that. This is from Sam Patnaik, the follow-up questions from NCATS. What is your database for these 1.2 billion compounds, and are these synthesizable?
Yeah, so that's a good question. So these all come from the Zinc database, and which is run by Brian Choquette and John Irwin. And these are all compounds that are make-on-demand compounds from enamine, basically. 90% are from enamine. And the success rate of synthesis is 80%. So not 100, it's not 100%, but it's 80%. So as I tell people, this is screening. It's not analytical work. So we're happy with 80% success rate is perfectly fine for us. So.
That sounds reasonable. Okay, here's another question that has high number of likes. What do you attribute the long-lasting antidepressant effects to? Do you believe the compound is still present at 14 days, or are there other effects going on?
Yeah, we don't know. And that's something we're trying to find out right now. It's a good question. And we're basically doing transcriptomics and proteomics studies at the single cell level, and to find neural populations in vivo to try to find out what's going on. We basically don't know what's going on. The compound is rapidly metabolized. We know that it's gone from the brain within a day. So it's not, and it has no reactive groups. But we think, we actually have seen this effect with some psychedelics. So DMT is novel in that it also has this really long-lasting effect. and humans as well have reported these sort of long-lasting beneficial effects of DMT, in particular for post-traumatic stress disorder. So we and others in the field are pretty convinced that there are some long-term neuroplasticity. There's a long-term neuroplasticity that occurs, but I would say that's sort of the big question, is what's going on right now, and I don't think anybody has a good clue on that. So, but it's a good question.
Fantastic, thank you. Next question here. During HTS, how do you identify false positives and false negatives? And a follow-up question to that.
Okay.
Yeah,
so
I'll get your answer.
We have orthogonal assays, basically. So a standard approach. We have orthogonal assays for everything. So
fantastic. Good to hear. And then the next one, question.
I've been doing small molecule screening since 1998, folks. So way before NCATs ever existed. I've been running the screening program since 1998. So
that's fantastic.
We have a lot of expertise in small molecule screening.
I bet. Okay. Oh, that's disappeared. So, okay, we'll take the other one. Okay. What's the conformation difference of the GPCR structures induced by the G-alpha beta arrestin biased compound you identified? Can you see from the solved cryo-EM structure or docked model?
So I would say the big disappointment is we haven't seen any big differences. So we have solved, people that follow my work, we solved the G protein complexes, we solved the arrestin complexes, and we solved the naked receptor complexes. And we don't really see any big differences in the conformation of small molecules. I mean, there are some small differences, but it's not, and you can, retrospectively come up with molecular dynamic simulations for bias, but it's been really, I would say, a disappointing exercise. We initially hoped that by solving all of these structures, we would really get a handle on how to create bias ligands, and that has not been very successful. Docking is very successful in that it allows us to get all these multiple chemotypes, some of which have bias in them, and it's not clear to us why they have this bias, but they do, and we can use them for further optimization. Instead, so I recently published a little sort of opinion piece on molecular glues as perfectly biased ligands. And basically what we found is for some receptors between the receptor and the G protein, there's another site there for small molecules to bind. And we have found that if we target that site, we can rationally design biased ligands because they're binding basically at the receptor transducer interface. And of course, those interfaces are different depending on if it's a G protein, arrestin, and so on. So that's probably the approach we're going to use going forward.
Makes sense. I have a note here. It says Greg Tawa asked this question about the huge chemical spaces because you asked. Greg Tawa is the person that asked the question about huge chemical spaces. Okay. And this is, by the way, this webinar is open to the public outside NCATS as well. So these questions are trickling in from all over the world as well as NCATS. It is exactly noon. We have plenty more questions. Do you mind if you stay 5 minutes over and we tackle a few more questions?
Sure, sure. And people can send me questions by email if they wish as well, or Twitter. I'm on Twitter,
That's awesome. Let's tackle one or two more and then we'll wrap this up because it's just there's so much excitement about your work, Bryan. So I want to get at least two more questions in. So have you considered potential inhibition of the HERG, which is crucial for cardiac repolarization for drugs that target?
Oh, yes. So
5-HT2A?
So every drug is tested for HERG. We have hERG assays through the PDSP. So of course everything is hERG negative. Everything has no CYP inhibition, no, all off targets, every off target, they're negative. They don't hit any kinases. They don't hit any other receptors, et cetera. These are really good compounds. So, but that's a good question, and I didn't mention it. So, but yeah, they're all cardiovascular safety is fine. Yeah.
And then one final one here. And sorry for everyone that we're going to miss some of these questions, but you can just ask Dr. Roth directly. I think people are interested to learn about your strategy. There's a lot of strategy questions. So I'll take this one because it has the highest like. Please shed more light on the strategy that you use to validate docking protocol.
Oh, yeah, that's key. So that's the key thing. So probably, I think what you should do is have a talk by Brian Roth on docking.
Okay.
But
it's a good recommendation.
I'll just give you, so what's typically done is retrospective docking campaigns are done. So for many of the targets we look at, it's already known, compounds already know that interact with the target. And so what's insisted in the docking campaign is that the docking mode and docking model is able to pick out known compounds that interact with the target and not pick out chemically matched decoys. So the Schoiket and Irwin group have really spent a lot of time basically making these libraries of what they call chemically matched decoys that have the same physical chemical properties as the compounds that are known to interact with the receptor. And so you do this receiver operator curve thing, ROC curve, and you look for enrichment of the true positives and de-richment of the true negatives. Basically, that's one of the things that's done. Of course, many other things, but that's sort of a key thing. Need to make sure that the docking pose and all the parameters that you use are able to at least recapitulate known compounds that interact with the receptor or target.
Awesome. One more, I promise, last one. From all the softwares you have used, which one is very suitable for analyzing the dynamics clearly? And that's been high.
So every person that specializes in molecular dynamics has their own little trick. I think Ron Dror basically uses all atom molecular dynamics simulations, and you can read his papers to find out what he uses. It's an interesting thing in the area of computational chemistry and computational biology that every group basically specializes in their own approach. So I'm a user. We just use what works. And we use basically the technologies that were invented by my collaborators. They seem to work. I can't make claims that they're better than anything else out there, but they work great. So we continue to use them.
Awesome, Brian. We'll stop here. Thank you so much for your time. There's plenty of more questions, but maybe we'll bring you back one day, next year or the year after, and, we can tackle more questions then. So thank you so much for your time. I appreciate you so much for a wonderful talk, the time you took to answer the many questions. Everyone, thank you for being here, attending. Sorry we couldn't get to all of your questions, but hopefully maybe we'll bring Dr. Roth back. So just a reminder for everyone listening here, next year we'll have Dr. Kathy High and Dr. Carolyn Bertozzi. So remember to join us to all the upcoming webinars next year. All right, everyone. Thank you so much. Thank you so much, Brian, again for being here. All right. Bye.